All right, we're going to go over Chapter 5, Energy and Life. The flow of energy in living things. So when we're talking about energy, it's really the ability to do work on a cellular level or otherwise. Energy is considered to exist in two states. There is kinetic energy, which is the actual energy of motion when we are in movement, and potential energy. Potential energy is stored energy that can be used for motion. All of the work that's carried out by living organisms is going to involve this transformation from potential energy into kinetic energy. And here you can see those represented. So the ball on the left has potential energy. It has the ability to move but is not yet moving, hence potential. The ball on the right has kinetic energy. As soon as that ball is in motion, that is kinetic energy because it is the energy of motion. And energy from the sun is captured by some organisms and then used to make molecules. These molecules are going to contain potential energy due to the arrangement of their atoms. And the potential energy in molecules is actually a form of chemical energy that can be used to do work in cells. Chemical reactions involve making and breaking bonds between atoms. When we're talking about thermodynamics, it's really the study of energy and heat changes. So there are many forms of energy, but really all of them can be converted to heat. And heat energy is going to be the most convenient form of energy for us to measure. But if we're studying energy or heat changes, that's thermodynamics. So energy is used and stored by these chemical reactions. And really, chemical reactions boil down to just the making and breaking of those chemical bonds. So whether we're making a bond, like here, where these two separate reactants have become this new product, or breaking bonds, like this reactant that has had a bond broken, and the result is these two products, either way, those are chemical reactions because we are making and breaking bonds. In the first law of thermodynamics, we're talking about what governs the energy changes of the universe, including those involved with any activity of an organism. So the first law of thermodynamics states that the total amount of energy in the universe remains constant. And energy can change from one state to another, but it can never be created or destroyed. So those are two points that are really supporting each other. We're saying we always have the same amount of energy, it's just changing form. And during the energy conversions, some of the energy is lost as heat energy. It's not destroyed, it's just lost from that system that it's in. Our second law of thermodynamics states that the amount of disorder, also known as entropy, in the universe is always increasing. And this is one of the most basic examples. This is from the book. But the idea is just basically that disorder and chaos, entropy, is always going to happen spontaneously without really any effort going into it. So even though it takes a lot of effort for us to clean our rooms and make them really organized and nice, it doesn't take any effort for us to notice that suddenly our bedroom is a state of chaos. And that's really just a simple representation of the idea that the chaos in the universe, this disorder, is going to constantly happen spontaneously. When we're talking about chemical reactions, the starting molecules of a chemical reaction are what we call reactants because they're reacting together, but also they are sometimes called substrates. The molecules that we have at the end of a chemical reaction are what we refer to as products. So a visual to help you guys remember which is just all of the ingredients on this side representing our reactants in a chemical reaction, and then the finished product being the cake. So really, we're talking about the same thing here when we look at these examples. In both cases, we are taking something and putting it together to make something new, or we could be taking things apart to create something new. But that's the basis of our chemical reactions and the bonds we're making or breaking. So, like weight. Reactants and substrates are the same thing? Pretty much. The difference is that we usually use the word substrate whenever we're talking about chemical reactions that are using enzymes. And we usually use the word reactant whenever we're talking about chemical reactions that don't use enzymes. So that's just to help you keep those separate. When we're talking about these chemical reactions, there are two kinds. We've got endergonic reactions, and in these reactions, our products will have more energy than the reactants. And that's because these reactions require an input of energy for them to even take place. In exergonic reactions, our products are going to have less energy than the reactants. And that's also makes sense because these reactions are able to occur spontaneously without any energy input. So here you can see a representation of both of those happening. In our first example, we've got endergonic reactions. You can see we've got our reactants and the energy that is associated to the reaction that's taking place. You can see we are higher energy with our products than we are with our reactants. And that's because we had to put energy into this reaction for it to even happen. So imagine this like reactants plus energy. And that energy stays with our products. So that's why our products have higher energy in endergonic reactions, because we've put energy into the reaction. On the other hand, our exergonic reactions are able to occur spontaneously because we don't have to put any energy in to make these happen. Just the presence of the reactants alone is going to cause the reaction to happen, and then our products will result, and we will actually release energy as we are creating products. So that's why our products are now lower energy, because the energy that was within the reactants is now been released as we have created our products. So think of energy as exiting an exergonic reaction. Here's something, another kind of visual to make that stick. So in endergonic reactions, our energy is going into the reaction to make something. Like how we have to put energy into moving this boulder up a hill. It's not going to happen spontaneously on its own. We have to make it happen. We have to supply energy. Exergonic reactions can occur spontaneously. You could think of it like an avalanche. You're not putting any effort into making it happen. You're not adding energy to the reaction. The reaction can occur spontaneously. And so examples of those are ATP breakdown, when we're breaking down that energy molecule ATP. We have energy going into the reaction, and that's why our products will have more energy than our reactants at the end of the reaction. And then we also have another example of an exergonic reaction where, like cellular respiration. In this case, we're not putting any energy in, but just the reaction that's happening between glucose and oxygen is creating an energy output. Here you can see an endergonic reaction. We have energy combining with our reactant to create our product. As a result, these products are going to have more energy because that energy wasn't released. It just went into the product. Our exergonic reactions. Here you can see our reactant coming in with other reactants, and then our product and the release of energy. So energy has exited from this reaction. That's why these reactants, what starts at the chemical reaction, is going to have more energy than the product because that energy was released as a result of the reaction. But they're constantly feeding into each other. So that is a cycle that feeds into each other. It is a coupled reaction. So you can see here we have our energy going in with our reactant to create this higher energy product. Here you can see a reactant combining with these other kind of byproducts of an endergonic reaction. They're going in together to create our product, and energy is being released at that same time. then that energy can go in with another reactant and create a new product. This is just a constant cycle that is going back and forth between each other. All of these chemical reactions are going to require an additional initial input of energy called activation energy. And the activation energy initiates the chemical reaction by destabilizing those existing chemical bonds. So basically making them ready to break. Reactions are going to become more likely to happen if their activation energy is lowered, because we're thinking of the activation energy like the start line. So the lower that start line is, the faster this reaction can start. And this process that we're talking about is called catalysis. So these catalyzed reactions are going to proceed much faster than any non-catalyzed reaction. And enzymes are what's responsible for catalysis. So you could think of enzymes as the catalyst that is lowering this activation energy and making a reaction faster and more efficient. Here's a picture to kind of represent that. So here we have a normal endergonic reaction where our product is going to have higher energy because energy was supplied. Here you can see our reactant in an exergonic reaction where energy will be released, so our product is going to be lower energy than our reactants. But really, both of them have this activation energy level, and this is the level that we have to get to for the product to be created. If we bring in an enzyme, we are able to lower that activation energy. You can see it was here. Once the enzyme is helping us, now it only takes this much activation energy to make this reaction take place. And so that's what's happening in catalysis. These enzymes are the catalysts. They're used by cells to perform particular reactions. And they will bind very specifically to a molecule and stress the bonds to make that reaction more likely to proceed. These enzymes have an active site, which is the site on the enzyme that binds to the reactant or the substrate. And they also have a binding site, which is the site on the reactant where the enzyme is going to bind. So really, we're just calling these are active and binding sites, but they're just different names because of what they're on. So where our enzyme is going to have a spot for the reactant, that's the active site. And where our reactant is going to have a spot to meet the enzyme, that's the binding site. This is to help make that message a little more clear. So we've got an enzyme. This is the active site, the spot available for a potential substrate to bind. And here the enzyme's saying, I'm here to make this reaction faster and easier. And the substrate's saying, oh good, I've been looking for my partner for so long. So the substrate is one piece, or one reactant of what could become a product. It's looking for the other piece to interact with that chemical reaction. But binding to this enzyme makes it way more accessible to whatever other substrate or reactant is looking to bind with it. So once that enzyme and substrate have combined together, we have an enzyme-substrate complex. The binding of a reactant to that enzyme is what causes the enzyme shape to change slightly. And this leads to an induced fit, where the enzyme and substrate are going to fit really tightly together as a single complex, that enzyme-substrate complex. The enzyme will lower the activation energy that's needed for the reaction so that it can happen more easily. And also, the enzyme is going to be unaffected by the chemical reaction. It can be reused. It's not like its shape has changed and it won't go back. As soon as that substrate has been released from the enzyme returns to its original shape and is able to be reused again for another chemical reaction. So the process of catalysis is the process where an enzyme comes in to help a chemical reaction happen faster and more efficiently. And here's a way you can see that happening. So think of this as our enzyme, this shape here. And then here are our substrates. They are looking for each other. They could float around for a really long time, looking to be able to be close enough so that they can create a bond with each other. But it happens a whole lot faster if an enzyme's present, because this has a strong affinity for the active site, and this substrate has a strong affinity for the active site. If they get pulled into those sites, now suddenly they're held so close together that it's really easy to form that bond they need to form. And now the product can be released. Because once these have formed a bond together, that has changed the chemical structure and the affinity to the enzyme itself, and that's what signals for the release of the product. Here's another example of a way we could represent those enzymes and the specific active site that is determining what reactant or substrate will be able to bind to it. Here's another picture showing really the same thing. I just wanted to be able to show it in a couple different ways with a couple different images. So here again, we have an enzyme. The active site is where a substrate can bind. Here, the reactants, the substrates, are coming to bind in their specific shaped spots. Once they are bound to the enzyme, we have an enzyme-substrate complex, and they're held so close together they can form the chemical bond that will change them into a single product. At that point, they will be released from the enzyme, and the enzyme can go perform more chemical reactions. Also, the chapter mentions the biochemical pathway, and this is just the idea that catalyzed reactions will occur together in sequence. So very often, the product of one reaction is going to be the substrate for the next reaction that needs to happen until a final product is made. So here you can see we've got a substrate binding to enzyme 1. Another substrate will bind until we have our product. That product becomes the substrate for enzyme 2 until another reactant or substrate binds and we have a new product, and so on until we have a final product. And that series of reactions with multiple enzymes producing a final product is called the biochemical pathway. But what you need to remember about it is that this product is becoming a reactant. This product is becoming a reactant. This product is becoming a reactant. So that's the whole idea of the biochemical pathway. Also, our enzyme activity is going to be affected by temperature and pH. Our enzymes function within an optimum temperature range, and when temperatures increase too much, the shape of those enzymes can change due to the denaturing or the breaking of the bonds of those protein chains. And that's essentially what's happening when we get a really high fever. We are denaturing proteins so much that our enzymes cannot function. And that also happens with pH range. If our pH is too off, it can also change our enzyme shape to the point where they are not able to function. Most of our human enzymes are going to work best within a pH range of 6 to 8, so that's pretty neutral. We know that 7 is totally neutral. And there are some exceptions, like the stomach enzymes, that are going to function in much more acidic ranges, but that's kind of what they're designed for. The majority of all enzymes outside of the stomach are going to need that 6 to 8 pH range for the majority of humans and other organisms. So here you can see the enzyme is saying, "My love, please, it's just a pH change, don't go." And here's our substrate saying, "No, darling, you've changed because this pH has changed the shape enough that now we don't have a spot where this substrate fits so nicely and has a really strong affinity to bind." So that pH change has changed the shape of our enzyme. Here you can see it's not a perfect number that every enzyme needs to exist within. It's more of a range for temperature and for pH. So here you can see this is the optimum temperature for human enzymes to function. And then here you can see the optimum temperature for the enzyme for a hot springs bacterium, which are going to want a much higher temperature to exist in, which makes sense. They're bacteria that live in hot springs. Here you can see the pH range that is optimum for pepsin, so super acidic. And then here's the optimum pH range for trypsin. So those are just some various examples of the ranges that can apply to these enzymes. Our cells can control our enzymes by altering their shape, and that can happen intentionally. So allosteric enzymes are going to be affected by the binding of signal molecules. We've got two different signals happening here in these examples. So some of our signals, our signal molecules, will act as repressors. And when we're repressing something, like you're repressing a memory, you're trying to stop that memory. So let that help you remember what the repressor is doing. Because our repressors will inhibit the enzyme when bound and make it to where they can't do their job. We also have activators. You could think of activists, like political activists that are trying to make things happen, that are trying to activate something. And so these signal molecules are changing the shape of the enzyme so that it can bind. Basically, the enzyme is useless for this purpose until the activator binds, and then bam, the reaction can take place. The substrate can now land on the enzyme because of that signal molecule that was an activator. And here you can see those happening, but I'd rather, instead of being kind of overwhelmed by looking at these side by side, let's look at them one at a time. So first, our repressors. You can see our enzyme here, and then this is the normal substrate that has an affinity and will bind to that enzyme to create products. But that same space that can fit our substrate can fit this other shaped repressor, a signal molecule. Except because of the different shape of that repressor, it's going to change the shape of the enzyme entirely once it binds. Now there's nowhere for the substrate to bind because the enzyme shape has changed. So that's how we are repressing this reaction. The substrate can't get access to the enzyme, so it's not going to be able to bind to something else to create a product. On the other hand, our activators, here's our enzyme, which has a shape where the substrate that is meant for it is not going to have enough of an affinity to bind to it until the activator lands in that spot and changes the enzyme shape just enough to allow for the substrate to bind. And now our product can be created. So think of the activators as activating that enzyme function. When we talk about feedback inhibition, it's a form of enzyme inhibition. So think of us stopping the enzyme. And it's where the product of a reaction is acting as the repressor. So the thing that was made in the reaction is now acting as a repressor. And in this situation, we have both competitive inhibition and non-competitive inhibition. During competitive inhibition, our inhibitor competes with the substrate for the active site. So they're both trying to land in the same spot. And the inhibitor can block the active site once it binds, so that now there's nowhere for the substrate to land. Non-competitive inhibition, the inhibitor will bind to the allosteric site, which you could think of as just a separate site on the same enzyme, and it's going to change the shape of the active site on the enzyme so that now the substrate can't bind at all. Both of these will stop the enzyme action from proceeding, but in different ways. So how enzymes can be inhibited. And remember, we're thinking inhibited means stopped. So first we have competitive inhibition. Here you can see something else has landed where the substrate was trying to land. That's what makes it competitive. They're competing for this same active site. But now over here, this is the allosteric site. So it's not the active site where the substrate's trying to land. It's a whole different site. And this non-competitive inhibitor can land here and change the shape just enough that now this will no longer fit in the active binding spot. So that has inhibited enzyme action. So here, this is just a little graphic to help us remember competitive inhibition. You can see the repressor protein has landed in the active site where the substrate was trying to land. The substrate saying, hey, that's my enzyme. And the repressor protein is saying, go away, the seat's taken. So that's what we mean by competitive. We're competing for that same active site. Versus non-competitive inhibition. This is the spot that would have been perfectly shaped for our substrate, except whenever this signal molecule bound to the allosteric binding site, suddenly our enzyme shape has changed and the substrate can't land there anymore. So just like with the pH change and the other example where we use this comic, the same thing's happening here, but it's instead of pH change causing the enzyme change shape, we now have an allosteric binding site that has changed the enzyme shape. And now our substrate can't bind. So once again, please don't go, we're made for each other. No, darling, you've changed because of this activity on the allosteric binding site. So that's our overview of enzymes and how they function. Now we need to talk about ATP. So this is our energy molecule. Whenever we get energy from the sun or from food sources, we have to convert it into a molecule that we can actually use. And this energy molecule is called adenosine triphosphate, also known as ATP. You can see the ATP is for adenosine, tri, and P for phosphate. That name really describes exactly what's in it. So here's our adenine. Every ATP molecule will have this ribose sugar. The adenine and the ribose combined is adenosine. And then we have 3 phosphates, 1, 2, 3, on that molecule. So that's the structure of our adenosine triphosphate, our energy molecule. And the phosphates on this molecule are negatively charged, and it requires a lot of chemical energy to keep them held together. So that means that the phosphates are really poised to come apart. They're like, oh, I put this here. This phosphate is ready to go. It's like, just say the word, and I will break this bond and give you the energy you need. Because when this bond breaks, that's when the energy is provided. ATP is the energy currency of the cell. And when the endmost phosphate group is broken off, like in that last example, energy is then released. So we have ATP. After the chemical reaction, we have ADP, which stands for adenosine diphosphate, meaning 2 instead of 3. And then we have an inorganic phosphate and the release of that energy that needs to be used. And then it's a whole cycle that just continues. So that phosphate bond breaking is going to free, so here we have ATP. The phosphate bond will break when energy needs to be supplied. Then we have ADP, which is adenosine diphosphate, basically the same exact thing, just missing that phosphate that was released here. And when that phosphate's released, that's when the energy is supplied. Once we have a floating inorganic phosphate with energy that's been released, we can create another molecule of ATP with our ADP. So this cycle just continues constantly. And that's how we're getting ATP into ADP, back into ATP with that constant energy release for all of our cellular function. So this is an example of coupled reactions, and also other chemical reactions could be happening at the same time as this ATP to ADP process. Because usually endergonic reactions are coupled with the breakdown of ATP, which makes sense because endergonic reactions, we have to put energy into them to make them happen. So a great place for them to exist is right where energy is being supplied by this breakdown of ATP. Also, more energy than is needed is going to be released by that breakdown of ATP. So at the same time that we are breaking that phosphate bond and releasing energy, we're also giving off heat. So our cells use 2 processes to convert energy from the sun and from food molecules that we've ingested into ATP. The first of which is photosynthesis for any photosynthesizing organisms. And in this, some cells will convert energy from the sun into ATP molecules and then use the ATP to make the sugars that will store potential energy. In cellular respiration, the other way we're converting this ATP so we can use it, our cells are breaking down the potential energy in sugars and then converting it to ATP. So photosynthesis, we have energy from the sun into ATP, and we're using it to make sugars to store potential energy. Cellular respiration, we are breaking down that potential energy so we can use it and convert it to ATP. And then this is just how our cells are using that ATP energy to power that cellular work. It's really everything you could think of, but these are specific examples that help you connect what we're using those ATP molecules for. So the first of which is biosynthesis. And in this, our cells are using the energy released from the exergonic hydrolysis of ATP to drive those endergonic reactions, like protein synthesis. And this is a coupled reaction. So it's an approach called energy coupling, where the energy is provided for an endergonic reaction, and then an exergonic reaction is producing more energy alongside each other in just kind of a cycle. Also, the contraction of our muscle cells. So filaments of protein are repeatedly sliding past each other in order to achieve a contraction of that muscle cell. And then an input of ATP is required to bring it back and reset so it can slide and contract again. So we are contracting the muscle, ATP is then supplied so it can go back to its original position and be contracted again. In chemical activation, we have proteins that can become activated when a high energy phosphate from ATP attaches. So like we said before, ATP is releasing this phosphate, creating ADP and the inorganic phosphate. And then once it attaches to specific proteins, it can activate them. And other types of molecules can also become phosphorylated by the transfer of a phosphate from ATP. On top of that, we've got importing metabolites. So we've talked about how we have ion pumps that allow us to get things in and out of the cell. And then we also have coupled transport proteins that allow things to come in if they're buddied up together. So here you can see our sodium is leaving the cell. It will couple with sugar to be brought back in. But that is still, even though we're not seeing ATP on this side of the process, this is only possible because of the ATP that let the sodium ions come out. So really, both the action of the sodium-potassium pump and anything that's being brought in through coupled transport proteins is being powered by this molecule of ATP that's going to break a phosphate bond and leave a phosphate on the sodium-potassium pump. ADP will float away to be used or remade into ATP. And because that ATP let sodium ions come out, now there are sodium ions available to bind with sugar and come through this coupled transport protein, bringing the sugars into the cell that we need. Here we've got active transport happening. This is a more in-depth example with our sodium-potassium pump, but most animal cells are going to want to maintain a low concentration of sodium ions inside. And they're going to want a higher internal concentration of potassium. So that's why we're constantly pumping out the sodium and bringing in the potassium. All of that is powered by the ATP that will break a phosphate bond and provide that energy to the sodium-potassium pump so it can change shape and then let sodium ions go out and then change shape and let potassium ions come in. Also, we talked about our long-distance intracellular transport. That's happening here. Remember, I told you guys we've got our microtubule that's acting like a train track. We've got our connector proteins, which are acting like the wheels of the train. We call them our motor molecules. And then we've got our connector proteins or connector molecules, which we're thinking of like the cargo cars of the train. They're connecting the moving parts to whatever needs to be transported, the cargo, the vesicle. So this cytoplasmic transport is within a cell's cytoplasm, and the vesicles are going to be dragged along those microtubular tracts using those molecular motor proteins, which are then attached to the vesicle with the connector proteins. And all of that is being powered by ATP and its effect on the motor proteins. That's what's allowing for that kind of walking motion of these molecules. We also have our flagellar movements. So for sperm or other cells that have a flagellum, the microtubules that are within that flagella slide past each other, similar to our muscle contractions, and that's what produces our flagellar movements. But ATP is powering that as well. It's powering the sliding of those microtubules back and forth. And then once again, we've got our cell crawling movements. And in this, our actin filaments inside the cell and in the cytoskeleton are continually assembling and disassembling to achieve changes in cell shape. And that's what's allowing our cells to kind of crawl over substrates or engulf different materials. And the dynamic character of actin is controlled by the ATP molecules. So every kind of cell movement we're describing has, it relies on this ATP energy molecule. So here you can see our heat production that's happening with this bee. And in the hydrolysis of ATP molecules, there's that release of heat. Reactions that hydrolyze ATP are often going to take place in the mitochondria or in contracting muscle cells, and they then can be coupled to other reactions. So then the heat that is generated by these reactions is what's used to maintain that whole organism's temperature. So the whole supply of heat, all of our cellular functions and cellular movements, it's all coming down to this energy molecule, adenosine triphosphate. And that's our summary of Chapter 5. We will go on to Chapter 6 next time, where we will talk more in depth about photosynthesis.